Hello students, welcome to Shri classes. Let us start the new chapter, second chapter in chemistry, acids, bases and salts. Chapter flow. First one. Acids and bases, their definition and types. Second one, measurement of strength of acids and bases. Strengths of acids and bases. Third one, acid base indicators. Acid base indicators. And fourth one, importance of pH in our daily life. Importance of pH in our daily life. Fifth one, chemical properties. Chemical properties of acids and bases. And last one, salts. Salts, properties, preparation, and uses, applications. This is how the chapter flow. So please note it down. I'll start with acids, bases, their definition, and what are the different types of acids and bases. Okay, so this is how the chapter flow is there. Here most important part is chemical properties of acids and bases. Of course everything is important, but mainly there are a lot of chemical reactions in chemical properties of acids and bases, and also in the salts, the preparation, applications, and some of the properties are very important. So these two are fifth and sixth is most important part of this chapter. Okay, so please note down the chapter flow. Now let us see the definitions of acids and bases. To define acids and bases, it has taken a lot of time and a lot of research by the scientist. Just like an atomic theory, atom, in 1808 atomic theory was built by Dalton. Dalton's atomic theory we have studied in 9th standard, that it was in 1808. But when the electrons were discovered and neutrons were discovered, if you look at electrons were discovered at the end of the 19th century, that is 1896. What about the protons? Around 1908 or 9, something like that, during that period. And neutrons were discovered somewhere in the 1932 by James Chadwick. What I mean by this is, almost it took a centuries to establish the atomic model and what are the contents of the atom. Similarly, to define his acids and bases, based on their particular taste and all, it is very easy. But scientifically, in order to define acids and bases, it has taken a great amount of efforts. If you study in higher studies, if you come across, the definitions of acids and bases are given by three famous, of course, four scientists, I'll say, like, Arrhenius theory of acids is there, and then Bronsted and Lowry theory of acids is there, then Lewis theories of acids. You must have seen Lewis dot structure. So Lewis theories of acids are there. So these are the three theories of acids which will come across in higher studies. But for 10th standard, we'll go by very simple definitions basically for the acids basis, and how to measure the strength of the acids and bases, what are the measurements, units of measurement, and how to define the classification, that is the types of acids. So in terms of the definitions, very primitive definitions are given for 10th standard. Anyway, we'll study whatever that is given in your textbooks. Okay, so first of all, let us start with the definitions of acids. Acids are those substances which taste sour. Okay, acids are those substances which taste sour and they turn blue litmus solution to red. What is that they do? They turn blue litmus solution to red, and these are the substances which give H plus ions as the only cations in aqueous solutions. They release H plus ions as the only cations in aqueous solutions, or H plus ions as the only positively charged ions in aqueous solutions. What I mean by aqueous solutions is these solutions, when you dissolve in water, they will release H plus ions. So for example, here I will take HCl. What happens in the presence of H2O for HCl? It will give you H plus ions plus chloride ions. Understood? In water, it will split into H plus ions and chloride ions. H plus ions are called hydrogen ions. Now, this H plus ions being unstable, it cannot remain as it is. It will again react with water. It will react with water to give H3O plus ion. H3O plus ion. And this ion is called hydronium ion. This is called hydronium ion. So, acids are the substances which release H plus ions as the only positively charged ions in aqueous solutions. In aqueous solutions means in the presence of water being the solvent there. Okay. So, these are the three ways in which acids can be defined. What are the examples of such acids basically? Let us take it. Sodium hydroxide, hydrochloric acid, HCl, H2SO4 and HNO3. Yeah, these are some of the examples for acids. Now similarly, how do you define the bases? Bases are those substances which taste bitter. They taste bitter and they turn red litmus solution to blue. Here red litmus solution or blue litmus solution, whatever I said, these are basically the indicators. They indicate the presence of acid or bases. So bases, they taste bitter, they turn red litmus solution to blue, and also in aqueous solution, bases are the substances which release hydroxide ions as the only negatively charged ions. Hydroxide ions are represented by OH minus ions. These are called hydroxide ions. Hydroxide ions as the only negatively charged ions in aqueous solutions. Which are the examples here? Sodium hydroxide, potassium hydroxide, calcium hydroxide, magnesium hydroxide, etc. These are the bases. Now again in bases you have one more thing, alkalis. ALKALIS. What are alkalis basically? Water soluble bases are called alkalis. These are basically water soluble. Water soluble bases are called alkalis. Which are the water soluble bases? Example, whichever I have listed over here, all these: sodium hydroxide, potassium hydroxide, etc. Sodium hydroxide, potassium hydroxide, all these are water soluble bases. Now these are water soluble bases. These are water soluble bases and called alkalis. If you ask me which are the bases that are not soluble in water, then we say insoluble bases, water insoluble bases. Water insoluble bases. Copper iron and lead oxides and hydroxides. These are water insoluble bases. For example, CuO, FeO, PbO, or CuOH twice, FeOH twice, and PbOH twice. All these are bases which are not water soluble. I hope you are able to understand over here. So how did we define acids? Acids are the substances which taste sour and they turn blue litmus solution to red, and in water when you dissolve the acids release H plus ions as the only positively charged ions in aqueous solutions. Example, hydrochloric acid, sulfuric acid, nitric acid. This is hydrochloric acid and HNO3 is a nitric acid. How do you define similarly bases? They taste bitter, they turn red litmus solution to blue, and in aqueous solutions bases are the substances which release hydroxide ions as the only negatively charged ions. Example, sodium hydroxide, potassium hydroxide, all these. Similarly, alkalis, you must know the definition. These are water soluble bases and examples are given. Sodium hydroxide is also called sodium hydroxide caustic soda, and this is called caustic potash. And water insoluble bases are also there. You must remember, copper, iron, lead oxides and hydroxides are water soluble bases. Okay, so I hope you have understood a simple definitions of acid bases. Now let us try to understand what are the different types of acids. Okay, the different types of acids and bases. Here we can classify these acids in simple types, very simple types basically. The first category can be organic acids and mineral acids. Okay, and second one, strong acids and weak acids, and third category can be concentrated acids and weak acids, dilute acids. These are very simple types. Okay, in organic acids, if you come across, what are these organic acids basically? These acids are derived from plants and animals, or these acids are naturally occurring in plants and animals. So naturally occurring in plants and animals. And weak acids, these are not strong, these are very weak acids. And some of the examples you can see here. Which are the examples for this one? Citric acid. Where do you find citric acid? In lime or lemon. Then lactic acid. Lactic acid you must have heard. That is in curd you will find lactic acid. Curd is also called sometimes sour milk. Acetic acid. See, vinegar is nothing but 10% acetic acid. That means acetic acid is present in vinegar, which we use it as a preservative. Then you have citric acid, lactic acid, acetic acid, tartaric acid, which is present in tamarind, malic acid, which is present in apple, and then oxalic acid, oxalic acid is present in tomatoes, and then you have one more thing is called formic acid. Formic acid is also called methanoic acid. Okay, this formic acid and methanoic acid is present in ant sting as well as in bee sting. You know, we get irritated whenever due to ants bite as well as bee stings and all. So during that time we generally apply, in order to neutralize, baking soda and all we'll apply. Anyway, we'll study those details. Formic acid, which is also called methanoic acid, and this is present in ant sting and bee stings. Ants sting and bee sting. And in orange you can find ascorbic acid, etc. So all these are organic acids which are naturally present in plants and animals. Those are derived from plants and animals, and those are weak acids, and these are not harmful. Generally these are not harmful. Okay, so these are the different types of organic acids. Let us see next, what are mineral acids? Mineral acids, as the name suggests, these are man-made acids manufactured from minerals present in the earth. So these are man-made and made from minerals of the earth, and generally very strong. Okay, and they are strong and cause severe burns. They cause severe burns. Even these are harmful under dilute conditions. Even under dilute conditions, these are harmful. Harmful even under dilute conditions. So which are the examples of mineral acids? Hydrochloric acid, sulfuric acid, nitric acid, etc. These are the mineral acids. So in one categorization of acids, one type of acids are organic acids and mineral acids. I hope you are able to understand the difference between the organic acids and mineral acids. Once again, these are derived from plants and animals, whereas these are the man-made and manufactured from minerals of the earth. These are weak acids. These are very strong acids which will cause very severe burns to human beings, severe burns to the skins and all. And all these are not harmful. You can find all these acids present in one or the other kinds of fruits and vegetables and etc. Whereas hydrochloric acid, sulfuric acid, HNO3, which are man-made, they are very harmful and cause severe burns and very strong. Of course, these acids find lot of applications in industries, medicines, drugs, etc. Okay, so organic acids and mineral acids we have studied. Now let us go for strong acids and weak acids. What are the strong acids and weak acids? We'll look into that. Second one, strong acids. I think let me erase here. I'll just erase this. Second category, under that, strong acids. See, how to define these strong and weak acids? Strong acids are the acids which undergo complete dissociation into ions. What I mean by complete dissociation is splitting up of ions, splitting up of into ions whenever they come in contact with water as a solvent. Therefore, we say the substances which undergo complete dissociation in aqueous solutions are called strong acids. So, strong acids means they undergo complete dissociations. complete dissociation in aqueous solutions. So they undergo complete dissociation to form ions basically. Here the dissociation is nothing but formation of the ions. So strong acids are the acids which undergo complete dissociation in aqueous solution into ions. I'll write like this: complete dissociation into ions in aqueous solutions. Okay, and thereby they produce more number of H plus ion concentration, higher concentration of H plus ions. They produce high concentration of H plus ions. H plus ions. Whenever I say H plus ions, you must also remember that it can be H3O plus ions also, hydronium ions. Why hydronium ions? In aqueous solutions, hydrogen ion is not stable. It will combine with water to form hydronium ion, and H plus ions are responsible for acidic character. Equally, I can say hydronium ions are responsible for acidic character, whereas hydroxide ions are responsible for basic character, isn't it? Similarly, so strong acids are the ones which undergo complete dissociation into ions in aqueous solutions, and thereby they produce higher concentration of H plus ions or hydronium ions. This is one way of defining this one. Which are the examples for this one? Strong acids. Strong acids are basically HCl, hydrochloric acids, H2SO4, HNO3, etc. What kind of ion formation takes place over here? For example, HCl in the presence of water, okay, it will get ionized into H plus ions and chloride minus ions. But thing is, H2O and H plus being H plus being unstable, it will combine with water molecule to form hydronium ion. So sometimes we will write HCl plus H2O, okay, gives you H3O plus ions plus chloride ions. Understood? So this is also we can write sometimes. So if you look at this solution, the solution contains only ions. The solution contains only ions. Remember this one, because in weak acids it is not the case. Here the solution contains only ions. Which ions? Hydronium ions and chloride ions, because it has undergone complete dissociation. Similarly, sulfuric acid H2SO4, okay, plus 2H2O, it will give you 2H3O plus ions plus sulfate ions. Sulfate ions is SO4 2 minus, isn't it? So 2 to the 4 to 6, yeah. So here basically if you look at only sulfuric acid, it will split into H plus ions, that is 2H plus ions plus sulfate ions minus 2 or 2 minus, you can say sulfate ions. So in aqueous solution, whenever it comes in contact with water, sulfuric acid will split into H plus ions and sulfate ions. But these two hydrogen ions being unstable, they will react with water to form hydronium ion. Therefore, we represent the reaction something like this: sulfuric acid plus 2H2O gives you 2H3O plus ions plus sulfate ions. So here also, if you look at this solution, it contains only ions. Why it contains only ions? Because sulfuric acid undergoes complete dissociation into respective ions. Similarly, nitric acid. So the same thing, the ionization whichever we are talking about over here, complete dissociation into ions. If we compare the dissociation process with weak acids, what happens? Let us see here. Weak acids. So weak acids are the substances which dissociate only partially. That means only partial dissociation of substances takes place into ions. So weak acids are those acids which dissociate into ions only partially, which only partially dissociate into ions. And thereby they produce less concentration of hydrogen ions, high concentration in case of strong and less concentration, and thereby they produce less concentration of H plus ions. H plus ions. Understood? So weak acids are those which only partially dissociate into ions. They do not undergo complete ionization, they do not undergo complete dissociation, but they only partially dissociate into ions. Very important. This is the main point with respect to strong acids and weak acids. Strong acids undergo complete dissociation into ions in aqueous solutions, whereas weak acids undergo only partial dissociation into ions in aqueous solutions. Into ions in aqueous solutions. Okay. Now, because of this partial dissociation, less concentration, less number of H plus ions are produced, or here also I can say H3O plus ions, hydronium ions. Lesser concentration of or a low concentration of hydrogen ions is produced. Now, what are the examples for this one here? Examples for this one is all these organic acids which we discussed are the examples for weak acids. You can say acetic acid CH3 COOH. Okay. So here how the splitting of takes place? CH3 COOH in the presence of water, you can find CH3 COO minus plus H plus ions. But thing is, here it does not undergo complete dissociation, and therefore you can also find over here, molecules of CH3 COOH and also molecules of water. Now H plus being unstable, it will get converted into H3O plus ions. So if you look at the solutions, basically the solution does not contain only ions, but the solution contains a mixture of ions as well as molecules here. For example, water and CH3 COOH acetic acid, these two are neutral molecules, whereas CH3 COO and hydronium ions are the hydronium and CH3 COO minus, these are the ions, whereas CH3 COOH and H2O are neutral molecules, and therefore the final solution contains a mixture of ions as well as molecules. Whereas here I have written only ions, meaning the substances, they undergo complete dissociation to produce ions. Here they undergo partial dissociation into ions. Hence you can find the resulting mixture will have both molecules as well as ions in the solutions. Okay. So this is about strong acids and weak acids. Now next we will concentrate on, let us look at concentrated acids and dilute acids. I hope you have understood over here, things are very clear. Okay. Next category I will take now. Third category, concentrated acids and dilute acids. Just to explain to you what is the concentrated and dilute, here I will take a beaker, two beakers I will take here. Okay. Water, say for example in both the cases 100 ml, and you are adding H2SO4 sulfuric acid. Here it is 25 ml and here H2SO4 5 ml. What will be the resultant solution here? Sulfuric acid 25 ml and so here sulfuric acid is only 5 ml. See, also remember this is a dilution process. The concentrated and dilute are related to the dilution process. Dilution means you have to dissolve acids into water and prepare some solutions. Now always we have to remember by like this, AAAW, always add acid to water. It is very important. always add acid to water. Always add acid to water. If you add water to acid, it will result into the explosion, and the person, the handler, may get injured. He may get the burns also. The reason being, the dilution process is highly exothermic in nature. It evolves a lot of heat, and therefore always we should add acid to water. Triple AW, always add acid to water. This is how we can remember also. Now we are talking about concentrated and dilute acids here. Here the relative percentage or the relative quantity of acid in this solution is more. Here the relative quantity of acid is less, and therefore concentrated and dilute is related to the relative quantity of acid present in aqueous solution, isn't it? So concentrated acid, it is an acid in which the relative percentage of acid is more. What is dilute acid? Dilute acid is the one in which the relative percentage of acid is less. That is the only difference. So here I can say, if I compare these two, it is subjective basically, because it is after all the comparison of acids and base, comparison of concentrated and weak here, based on the relative quantity of acid present in aqueous solution. Now this 25 ml is present here. Here only 5 ml. Therefore this is a concentrated acid and this is a dilute acid. So I can write here concentrated, and here it is dilute acid, because it depends upon the relative percentage or the relative quantity of acid in aqueous solution. I hope you are able to understand this here. So how do you write it? So it is an acid, I can say like this: concentrated acid, it is an acid containing acid that contains relatively higher percentage of acid in aqueous solution. So similarly, dilute acid, or here I'll write dilute acid. It is an acid that contains relatively lower percentage of acid in aqueous solution. Whenever we refer to aqueous solution, remember that the solvent is water here. The major component of the solution being water, we call that solution as an aqueous solution. So dilute acid and concentrated acid, I think you have understood. Generally the concentrations are measured in moles per liter. All these concentrations are measured in moles per liter. Moles per liter is the measurement of concentration. So if you look at the dilute acids, the concentration or generally the acid present will be less than 1 mole per liter. Acid present is less than 1 mole per liter. You understand? In that case we say it is a dilute acid. So we have seen the different the definition of acids and the different types. The types in the sense, it is a very simple classification of acids. Okay, acid classification is totally different, but for our understanding we just divided acids into strong acids and weak acids, concentrated and dilute acids, organic acids and mineral acids. Okay, I hope you have understood the acids and their classification. Please remember, very important, in acids H plus ion concentrations are responsible for acidic character, whereas OH minus ions, which are called hydroxide ions, are responsible for basic character. Okay, fine. So now let us start studying the bases, that is the classification or the types of bases. Next is bases. Once again, what are the bases? Those substances which taste bitter, those substances which give hydroxide ions as the only negatively charged ions in aqueous solutions, those substances which turn red litmus solution to blue color. So these are called, and what are the examples? Sodium hydroxide, calcium hydroxide, magnesium hydroxide. Metal oxides and hydroxides are generally basic in nature. So what are the bases basically? Metal oxides and metal hydroxides. Okay, fine. Now in bases we studied alkalis also. What are alkalis? Alkalis are water soluble bases. Water soluble bases. Now how do you classify here? Here you have, see now instead of bases I am considering as alkalis only. We are talking about alkalis now. Strong alkali and weak alkali. Strong alkali, weak alkali, and another classification here is it is concentrated alkali and dilute alkali. Concentrated alkali and dilute alkali. So what is strong alkali and weak alkali? Let us study now. So an alkali which undergoes complete dissociation into ions in aqueous solution is called strong alkali. So a substance or a strong alkali is an alkali which undergoes complete dissociation into ions in aqueous solution. So strong alkalis are the alkalis, or strong alkali is an alkali that undergoes complete dissociation into ions in aqueous solution, and thereby it produces high concentration of which ions? High concentration of OH minus ions. OH minus ions are called hydroxide ions. Understood. So what is the example here? In this case, strong alkalis, sodium hydroxide and potassium hydroxide. These two are very strong alkalis, and the alternate name for this is, and this is caustic soda, the common name or a commercial name, caustic soda, and this is caustic potash. Okay. Now if you ask me what is this complete dissociation, see sodium hydroxide in the presence of water H2O, it will ionize into sodium plus ions plus hydroxide ions. You understood? This is how sodium hydroxide will undergo to produce sodium plus, it will undergo dissociation into Na plus and OH minus ions. OH minus are hydroxide ions, and remember the hydroxide ions are responsible for basic character. Similarly, potassium hydroxide in the presence of water, okay, K plus OH minus ions. Okay, these are the ones which are strong alkalis, and this is the example, and this is the ionization process, that is the complete dissociation into ions. Now let us look at weak alkalis. So weak alkalis, how do you define? Those are the substances which undergo only partial dissociation. You understand? They undergo only partial dissociation into ions. So undergo, substances that undergo, these are the alkalis which undergo only partial dissociation. Partial dissociation into ions. They undergo only partial dissociation into ions. So when they undergo partial dissociation, the number of hydroxide ions produced are lesser in concentration. Thereby they produce less concentration of hydroxide ions. Okay, which are the examples for weak alkalis? Examples are calcium hydroxide, magnesium hydroxide, ammonium hydroxide. NH4 OH. Okay, these are the weak alkalis. Also you must remember the resulting solution. Whenever this calcium hydroxide solution is dissolved in water, you find the resultant mixture will contain the ions as well as molecules. Which are the ions here? Calcium ions and hydroxide ions. And which are the molecules? Both calcium hydroxide molecules as well as water molecules. Very important, these things. So the resultant mixture in case of strong alkalis will have only ions, because it undergoes complete dissociation. The resultant solution, or the solution, if you look at over here, it will have a mixture of both ions as well as neutral molecules. Okay, so this is the difference between strong alkali and weak alkali and definitions over here. Okay, now let us study concentrated alkali and dilute alkali. Next is concentrated and dilute alkali. Similar to acids we studied, the concentrated and dilute depends upon the relative percentage of alkali present in aqueous solutions. If the relative percentage of alkali present in aqueous solution is less, it is called dilute. If it is present more, it is called concentrated. So, for example, if we want to prepare sodium hydroxide solution, same if you take 100 ml of water here, 100 ml of water, same here also 100 ml of water, and you put about 25 grams of sodium hydroxide crystals, and here only 5 grams of sodium hydroxide crystals. So the resultant solution, okay, which is a concentrated, the one which contains relatively higher quantity of alkali, is called the concentrated solution. So this is a concentrated solution, whereas this is a dilute solution. Here it is concentrated and dilute. So once again, the concentrated and dilute depends upon the relative percentage of alkali present in aqueous solutions. So how do you define this concentrated alkali? It is an alkali that contains relatively higher percentage of alkali in aqueous solutions. How do you define dilute alkali? It is an alkali which contains relatively lower percentage of alkali in aqueous solution. Okay, here again the percentage of alkali, if you look at the concentration, if you look at, it is less than, in dilute alkali, the concentration of alkali present is less than 1 mole per liter. The concentration is less than 1 mole per liter. So whenever the concentration is less than 1 mole per liter, those things are referred to be the dilute alkalis here. Similar to the acids, where we found the same concentration of acid, if it is less than 1 mole per liter, it was called dilute acid, similar to that one. So this is how we have seen now acids, bases, their definitions, and a small classification of acids, and a small classification, very simple classification of bases. Okay, so this constitutes the first part, and next we are going to study the measurement of the strength of acids and bases, and then the different indicators. Okay, fine. Now you will be directed to the PPT. You can make a detailed notes of that. Okay. So here you have a chapter flow, and you can pause the video and note it down all the time. Next comes acids, bases, their simple definitions and terms. Whichever I have explained, all these things are highlighted. It is preferable if you wrote down all these details, because from the textbook I have segregated everything and taken and represented in the form of PPT with all detailed information. It is preferable if you write all these details. Then comes acid types and the classification. Note it down. And then bases, alkali types. Okay, so please note it down. Now you must know very clearly H plus ions are responsible for acidic properties, and H plus being hydrogen ions being unstable in nature, they react with water to produce hydronium ions, and the formula for hydronium ions is H3O plus. Okay, this is called hydronium ion, and this is responsible for H plus, or this is responsible for acidic character, or it makes the substances acidic in nature. Similarly, hydroxide ions, these are called hydroxide ions, not hydroxyl. Hydroxyl is a group where OH is a neutral molecule there, but here it is an ion, and therefore we call this as hydroxide ion. And this hydroxide ion is responsible for exhibiting the basic property or alkaline property. Okay, so now let us look at how to measure the strength of the acids and bases. In what way we measure the strength? When do you say it is a strong acid? When do you say it is a weak acid? There must be some measurement, no? When do you say it is a strong alkali or a weak alkali? Some measurement is required for that one. So what is the measurement of the strength of acids and bases? So measurement of strength of acids and bases. Measurement of strength of acids and bases. Now, pH scale. P is small letter, H is capital letter here. pH scale is used to measure strength. pH scale is used to measure the strength of acids and bases. In this pH, P represents potence. This is a German word. Potence means power. So finally, pH means power of hydrogen ion concentration, power of H plus ion concentration. So the measurement of hydrogen ion concentration gives you the measure of acids and bases. That means, what is the concentration of hydrogen ions present in aqueous solutions? So how many numbers of H plus ions are present? That is measured in moles per liter. The concentration is measured in moles per liter. So that particular measure of hydrogen ion concentration in aqueous solution gives you the measurement of acids and bases. So what is this pH scale? Basically we'll study. Now one more important thing is, let us define the pH. What is pH? pH is defined as the negative logarithm to the base 10 of hydrogen ion concentration. So let us define pH, and then I'll take you through the scale which is from 0 to 14 and how to read that scale. Okay, so here definition of pH. Define pH. I said a pH scale is used to measure the strength. In that, P is potence, which is power. So the power of H plus ion concentration, or indirectly it is a power of hydrogen ions, meaning the strength of H plus ions indicates the strength of acids and bases. Now define pH. How do you define pH? See, pH is defined as negative logarithm to the base 10 of H plus ion concentration. So anyway, logarithms have not been introduced to 10th standard. For the time being, you understand only the definition of this one. No need to go much in detail. So how do you define pH? See, pH is defined as the negative logarithm to the base 10 of hydrogen ion concentration expressed in moles per liter. That is also very important, because the concentration of chemicals, basically the reactants and products, is measured in moles per liter. Concentration is measured in moles per liter, and here the concentration of H plus ions is expressed in moles per liter. Generally, the concentrations are written in square brackets. For example, what is the concentration of calcium hydroxide? This is how we write it. What is the concentration of hydroxide ions? So the square brackets generally represent the concentrations which are expressed in moles per liter. Fine. So once again, how do you define pH here? pH is defined as the negative logarithm, log means logarithm, negative logarithm to the base 10 of hydrogen ion concentration expressed in moles per liter. Once again, pH is defined as the negative logarithm to the base 10 of hydrogen ion concentration expressed in moles per liter. Now you must understand very clearly, when I say H plus ions, equally it can be H3O plus ions, which is nothing but hydronium ion concentration. So also I can say it is a negative logarithm to the base 10 of hydronium ion concentration, which is expressed in moles per liter. So I hope you have understood what is the pH. The pH scale we'll see still, but you have understood what is pH here and the definition of the pH. Now let us look at the pH scale and how it is measured on the pH scale, acid basis, how it is measured. Okay, let us look at this slide. These things have been explained already. Let us look at the pH scale. How does it look like? Here you can see one rectangular block, and 0 on one side, 7 at the middle, and 14 at the other side. So pH is basically a number which varies from 0 to 14. If pH is 7, then the substance is said to be in neutral condition, meaning H plus ion concentration and OH minus ion, H plus ion concentration and OH minus ion concentrations will be equal in that substance, and therefore the pH is said to be 7, and which is neutral. If the pH number is less than 7 up to 0, then acidic nature of the substance increases. If pH number is above 7 up to 14, then the basic nature of the substance increases. So from 7 towards the left side, acidic nature increases, and from 7 towards the right side up till 14, basic nature increases, meaning smaller the number, pH number, more acidic is the substance. Higher the pH number, more basic is the substance, or alkaline is the substance. So we have understood the reading of pH from 0 to 14. Once again, 0 indicates highly acidic in nature, the substance is highly acidic, and if the pH number is 14, then it is highly basic or highly alkaline is what we say. Maybe sodium hydroxide and all, you may get 14 pH. Let us look at this blue region over here. This is one part, blue region, and another is white part over here. What is this indicates? The blue region indicates hydrogen ion concentration. If I look at the scale, here the hydrogen ion concentration is very less and it goes on increasing, and it is highest over here. Hydrogen ion concentration increases towards the left side here, and therefore left side is highly acidic. Coming to hydroxide ion concentration, here to here, the white region, if you concentrate, hydroxide ion region is slowly and gradually increasing like this. Here it is maximum, here it is less, meaning hydroxide ion concentration is maximum over here. Therefore, if the pH is 14, then the substance is highly alkaline or highly basic in nature. So at 7, hydroxide ion concentration and hydrogen ion concentration will be equal. Therefore, the substance, neither it shows acidic nature nor basic nature, and the substance is said to be a neutral, and the pH value for the neutral substance is 7. I hope you are able to understand this one. Now, one more small thing. If the pH is highly acidic, pH is 0 indicates highly acidic, What is the concentration of hydroxide ion? We know definitely for a highly acidic substance, H plus ion concentration is very high. What about hydroxide ion concentration? Can you find hydroxide ions in that one? Maybe if present, hydroxide ion concentrations will be very less because H plus ion concentration is very high. Similarly, here if you look at, if the pH number is 14, then it is highly basic in nature. Do you find H plus ion concentrations? Definitely hydroxide ion concentration is very high, but H plus ion concentration, maybe if at all present, it is very negligible, which is very less. So overall, the measurement of H plus ion concentration can tell you whether the substance is acidic or basic only with respect to H plus ion concentration. Look here, more the concentration acidic, very less is the concentration of H plus ions, it is basic in nature. So this is how we read the pH. Once again, for your information, the pH values I am taking now. pH value is 0, highly acidic, 7 neutral, 14 highly basic. Smaller the pH number, acidic nature increases, highly acidic or acidity increases. Bigger the number, above 7, basicity increases, alkaline nature increases or basic nature increases. So this is all about the pH scale, and generally pH scale is the one which is used to identify whether the substances are acidic or basic. And in next slide and all, we'll see the indicators which are used to detect whether the given substance or the solution is acidic or basic. So I request you to note down all these contents which are very, very important. Now we'll study acid base indicators. What are indicators? These are also called acid base indicators. These are also called acid base indicators. As the name suggests, these are the substances which indicate whether the given solution is acidic or basic based upon the change in color or also based upon the change in smell. That means the substances which detect or which indicate the presence or absence of an acid either by a change of color or by a change of smell are called acid base indicators. So generally, how do you classify these acid base indicators? See, the acid base indicators are classified as common acid base indicators, common acid base indicators, and second one, these are universal indicators, and third category is olfactory indicators. Olfactory indicators. These are the three categories. Common acid base indicators and universal indicators, they show the change in color to indicate whether the given substance is acidic or basic, and olfactory indicators change their smell to indicate whether the given substance is acidic or basic in nature. So what are common acid base indicators? In common acid base indicators, you have there are two categories. One is natural indicators, and also you have synthetic indicators. Synthetic indicators. What are natural indicators? Substances which are naturally occurring and which indicate whether the given substance is acidic or basic. Such substances are called natural indicators. Example here are litmus. If I talk about the litmus solution is basically a purple dye, and this purple dye is extracted from litmus is here itself. I'll write here litmus solution. It is a natural indicator. This litmus is extracted from, it is basically a purple dye, and this is extracted from a division of plants called Thallophyta, and in that it is, the name of the plant is basically lichens. Fungi and algae living together in a symbiotic relationship, those are called lichens basically. So these are purple dye is extracted from lichens, and this belongs to a division Thallophyta. Okay, and litmus solution is purple in color. Okay, so next indicator, red cabbage leaves. Turmeric, or even some petals of flowers, colored flowers, petals of flowers of plants. Which type of plants? Plants belonging to, there are three categories of plants. The petals of the flowers which belongs to a particular three categories of plants, and these are hydrangea, it is called hydrangea, hydrangea. So it is a particular plant, and the flowers of which, the petals of flowers of which act as acid base indicators, hydrangea or petunia or geranium. Yeah, so these are the different types of flowering plants: hydrangea, petunia and geranium. And the flowers, the petals of the flowers of these plants would be able to indicate by changing their colors whether the given substance is acidic or basic in nature. So turmeric also, turmeric, one example I'll give you. On a white cloth, if a drop of curry falls, whenever we wash, when we apply soap, that stain will turn into reddish brown color. The reason is the presence of turmeric in curry. So the turmeric in curry turns into reddish brown whenever it comes into contact with soap solution which is basic in nature. So if the substances are basic in nature, turmeric changes its colors to red color or reddish brown. After washing, after rinsing with a lot of plenty of water, once again it will go back to yellow, which is the original color. When the turmeric goes off, then the stain changes back to yellowish color. Otherwise it will show reddish brown in color. So natural indicators are naturally occurring substances such as litmus. Litmus is a purple dye extracted from lichens that belongs to a division of plants called Thallophyta. And red cabbage leaves, turmeric powder, or petals of flowers of some plants such as hydrangea, petunia, geranium. These are the natural indicators. These are the substances which indicate the presence or absence of acids or bases by changing their colors. Now let us talk about synthetic indicators. Here you have methyl orange, and also you have phenolphthalein. Methyl orange and phenolphthalein are synthetic indicators which are manufactured basically. Okay, so these are common acid base indicators. Now what are these universal indicators? Universal indicators are mixture of natural indicators or mixture of common acid base indicators. Universal indicators are mixture of common acid base indicator. These are the mixtures of common acid base indicators. Generally, they mix the synthetic indicators rather than these natural indicators. They mix the synthetic indicators to produce universal indicators. So, why? I mean, what is the difference between these common indicators and universal indicators? Please remember, these substances will indicate the presence or absence of acid, but by changing the color. But these substances will not give you the value of the pH of a particular acid or a base. That means these substances will not give us the measure of pH, or the strength of pH is not indicated. For example, if you take the litmus solution, generally for acids, the blue litmus solution will turn red. Now if I take acids of pH 1.8 and pH 2.6, these are the two acids here. If I use litmus solution, a blue litmus solution will turn to red. Blue litmus solution will turn to red. But it only indicates the presence of acid in this substance. Meaning, the solution, litmus solution, will not give you the reading or the value of this pH at all. It will not give you. But there is a difference with universal indicator. What is this universal indicator? Being the mixture of common acid base indicators, especially the mixture of some synthetic indicators, they will change their colors. The universal indicators will change their colors to different colors, different combination colors based on the pH value there. That means the universal indicators would be able to give pH range for a particular solution given. Are you able to understand? For a particular solution given, if it is acidic or basic, whatever it may be, the universal indicator will give you a particular pH range. You understand? For example, pH range is 7.2 to 7.6, this color. 7.2 to 7.6, this color. So, by using universal indicators, if we get that particular color, a particular color, we have to match that with the given reference colors and identify the range of the pH. Understand? So, the main difference between common acid base indicators and universal indicators is that these common acid base indicators only indicate whether the given solution is or a substance is acidic or basic by changing the color. They will not give you the strength. Here, as I explained, even at the 1.8 pH, the blue litmus solution will turn to red. With even 2.6, blue will turn to red, isn't it? But it will not give you the value of the pH or the range of the pH. Whereas universal indicators give you the range of the pH. This is the main difference. Okay. Now, how these see, the universal indicators, the best example is a pH paper. pH paper. What is this pH paper? This pH paper means a paper which will be impregnated in universal indicator solution and dried up. And that pH paper, if you put into a test tube or a solution containing acid or base, it will change to a particular color. Now you will be given a reference chart in which the different colors along with the pH range will be given. We have to match the color obtained with the reference colors. You understand? And thereby we should be able to conclude fairly well the range of the pH for a particular substance. So that is the main difference between common acid base indicator and universal indicator. Okay. Anyway, we will look at your textbook diagram also, where a universal indicator gives you a different colors for different pH range and for different substances. We will see that one. Next, what is left in this one? Common acid base indicator, universal indicator, and one more is olfactory indicators. Third one, olfactory indicators. See, olfactory indicators are the indicators which change their odor or which change their smell and thereby indicate the presence or absence of acid. Say for example, we have onions, we have vanilla, vanilla, onion, clove oil. These are all olfactory indicators. Onion, vanilla essence, basically clove oil, etc. These are the ones. Now in basic solution, they lose smell, whereas in acidic they retain their smell. And this is how all these will be able to indicate whether the given substance is acidic in nature or basic in nature. I will also discuss the olfactory indicators with an activity given in your textbook. Anyway, we are going to discuss about 2-3 activities which are given, but before that you must understand the different types of indicators. I hope you are able to understand here. Even though only the names have been given in your textbook, but whether they retain or they lose smell, it is not given in your textbook. But last year there was a question based on, I think, this vanilla essence, whether it retains its smell or it loses its smell. The question had appeared. So you must note down all these things. Anyway, I will direct you to the PPT. Only in that case you can note it down. But for your information, remember, onion, vanilla, clove oil, in basic solutions they lose their smell or odor. In acidic solution they retain their smell and odor. Okay, what else is to be discussed? Now we can have a look at the PPT and then you can note down the details. Here, indicators, classification, and these things very important. Lose olfactory indicators, lose smell in basic, retain smell in acidic, which are all these are important points. You have to note it down. Here I have also given this the colored petals of some flowers such as hydrangea, petunia, and geranium. Try to remember these plants also. I know those are important. You may get question from any of these things. Okay, methyl orange, phenolphthalein, etc. Okay, fine. I hope you have noted down. Now I'll discuss some synthetic indicators and what is the change in color in the presence of acid or base, which is important. Here, couple of things are very important. See, indicator, I write here indicator, and neutral, acid, and base. I am going to write here the change in color that we observe of these indicators. I think I have to fill the ink. Let me check up now. Indicator, we'll use first one, litmus solution. Second one, phenolphthalein. Methyl orange. These are very important. Litmus solution is purple in color under neutral condition. Phenolphthalein is colorless, and methyl orange is orange in color. This is in neutral condition. You understand? Now, in acidic condition, the litmus solution changes to red, and in base, the litmus solution changes to blue. Next, phenolphthalein, in acidic, it remains colorless, and in base it changes its color to pink. Methyl orange, it is neutral, under neutral condition orange in color, and it changes to pink in acidic condition and yellow in case of base here. So these are the different changes in color that we observe for litmus solution, phenolphthalein and methyl orange. Okay, and apart from this, olfactory indicators also I'll indicate, that is onion, vanilla essence and clove oil. Generally these are the ones which are used, vanilla essence, these are the ones which are used to indicate acid bases. Onion, vanilla essence and this, all those, they retain their smell, retain smell, and under basic conditions lose smell. So apart from this, let me check if something is there. We have here in between, let me try to write that one. You may not expect a question from that, but these are definitely more important. I am adding one more synthetic indicator, methyl red here. Phenolphthalein, methyl orange and methyl red. So methyl red will change from red, it will be there, yellow to red. It is red, yellow to red. See here, methyl red, in neutral condition it is red in color, and it will change to yellow in acidic and red in basic. Okay, see here one chain you can see, phenolphthalein colorless to pink, pink to yellow, yellow to red. This is how you should be able to remember phenolphthalein, methyl orange and methyl red. What is that I said? In acid base, if you look at, see their natural color you will come to know. Phenolphthalein is colorless, you must know. Methyl orange is orange, methyl red is red in neutral condition. But when we talk about acid and base, see the sequence here, color to pink, okay, and then comes pink, pink to yellow, and then comes yellow to red. This is how you should be able to remember. Color to pink, pink to yellow, yellow to red. And it, this is the sequence which you should be able to remember. So these are the common acid base indicators which will not give you the pH range, but definitely would be able to tell whether the given substance is acidic or basic based upon the change in color for these indicators, and based upon the retention or loss of smell by these substances in the presence of acid or base. Okay, yeah. Now let us have a look at the pH paper in your textbook that is given. Here I have explained the same thing which you can note it down. Common acid base indicators, litmus is important, methyl orange, phenolphthalein, but the order in which to remember the order, you follow whatever I had written on the board. That is more important, because I go by the order there. First is litmus, phenolphthalein, methyl orange, methyl red. So that remembering to colorless to pink, pink to yellow, yellow to red, you will be able to remember that, and therefore you can note down the details from the board itself. Okay, and here this is a pH paper. What is this pH paper? It is a paper which is impregnated in universal indicator, and it is dried, and therefore there are different color combinations which you can observe over here. And this is a pH value 0, pH value 14, highly basic, highly acidic. So these are the different color combinations. So such papers you have to take it. It will be a paper basically. That paper, if you dip inside the solution given to you, and it will turn to a particular color. That color you have to match with this strip. Once if you match the color, and you should be able to read the pH values of those pH value, and by comparison you will be able to fairly judge the pH range as well as pH values. Okay, here gastric juice gives you this color. If I use pH paper having a pH about 1.2, whether it's a gastric juice or whatever it may be, having pH value approximately 1, it will show this color. If pH value is 11, it will show this color. If pH value is 14, it will show this color. Or if this color you obtain in a pH paper, this is 14. If this color is obtained, it is 7. So important of this is water and blood. See, the pH paper turns green. Please remember, this question had appeared, and also it is there in your exemplar. What is the color of the pH paper in blood? What is the color of the pH paper in water? The color of the pH paper in pure water and blood is green here, 7, green, and it is approximately neutral in nature. Okay, so that is important. Gastric juice 1.2 pH, lemon juice 2.2, pure water, milk of magnesium is magnesium hydroxide, and sodium hydroxide highly acidic, highly basic in nature, which is 14 pH. Okay, and this part also you note down the difference between common acid base indicator and universal indicator, okay, which I have explained already. Fine, let me check what else is there next. Now, very important, there is an activity 2.1 in your textbook. Activity 2.1. See here in your textbook there is an activity 2.1 which is given. So here you can see activity 2.1. I will read this activity very quickly. I hope I am also visible. Collect the following samples from science laboratory: hydrochloric acid, sulfuric acid, nitric acid, acetic acid, sodium hydroxide, calcium hydroxide, potassium hydroxide, magnesium hydroxide, and ammonium hydroxide. Put a drop of each of the above solutions on a watch glass and test with a drop of following indicators as shown in the table 2.1. What change in color did you observe with red litmus, blue litmus, phenolphthalein, methyl orange solutions for each of the solutions taken? Tabulate your observations. So the answer is given here. Activity 2.1. Hydrochloric acid, sulfuric acid, these are the samples taken. These are the solutions: red litmus, blue litmus, phenolphthalein, methyl orange. Understood? So the color observation, see, we have already studied this. You should be able to answer. Hydrochloric acid, sulfuric acid, nitric acid, these are acids. Acetic acid, this is also acid but weak acid, it's okay, no problem. Sodium hydroxide, calcium hydroxide, potassium hydroxide, magnesium hydroxide, all these are bases, isn't it? So red litmus in acids, red litmus will remain red only. Why? Blue litmus will change to red color, red litmus remains red color, or the purple solution, litmus solution naturally is neutral is purple, so purple also may change to reddish color. So important, red litmus solution no change, blue litmus they turn to red in acids. What about in bases? Blue litmus no change, whereas in here the red litmus changes to blue color, red litmus solution changes to blue color in bases, no change in acids. Blue changes to red in acids, no change in bases. Phenolphthalein for acids colorless to pink in bases, isn't it? Phenolphthalein is colorless in acid and changes to pink in bases. Methyl orange, it is pink to yellow, orange, pink to yellow, you understood? So in acids, in neutral it is orange, but in acids it is pink, in bases it changes to yellow. So this is the observation given. Next, activity 2.2. See, very interesting activity 2.2. I'll read it here. I'll just explain. See, your textbook gives the activity 2.2 something like this. You have to take some onions, chop it, fill those onions into a plastic bag, and also few pieces of cloth inside that bag, and tie it and keep it in the refrigerator for about 8 to 10 hours. Next day morning, you take out those piece of cloth, and what we have to do is, on one piece of cloth put acids, and another piece of cloth add sodium hydroxide. That is sodium hydroxide and acid. One piece of cloth HCl, we have to add HCl, and another cloth we have to add sodium hydroxide. Leave it for some time. Then both you can rinse in water. It should be rinsed totally in water. Now we have to smell these things, the cloth pieces. Surprisingly, wherever you had put acid, you can still find the smell of the onion. Just by rinsing the cloth with water, the smell cannot go. In acid, it will retain. Whereas wherever you on that piece of cloth where you put sodium hydroxide and rinsed with water, you will not find the smell of onion at all. Meaning, the olfactory indicator onion retains its smell in acid's presence and loses its smell in basic presence or in basic substances. So that is what the observation here we have written. Also, the same activity 2.2 discusses with vanilla essence and clove oil. The same observation. What we have to do is, in a test tube, take a few ml of vanilla essence and add hydrochloric acid to one test tube. In another test tube containing vanilla essence, you add sodium hydroxide. After some time, if you smell the one which has got acid, there you can still find the vanilla essence smell, whereas in sodium hydroxide solution, it will you will not be able to smell the vanilla essence smell. It will lose the smell there. Similarly, clove oil. In say few drops of clove oil you can take in a test tube, add a few drops of hydrochloric acid, and after some time you try to smell it, you will still find the clove smell. Means in acidic presence, it will retain its smell. Similarly, a test tube containing few ml of clove oil, if you add sodium hydroxide solution, few drops of sodium hydroxide, after shaking well, after some time, you will observe that you will not be able to see the smell of the clove oil. Means it has lost. So important thing is onion, vanilla essence, and the clove oil. All these things, which are basically olfactory indicators, they retain their smell in acidic presence, whereas in the basic presence, they lose smell. They lose their smell. All these are very important thing. Let me check one more activity I think I have discussed here. Yeah, approximate pH values of some common substances you can see. What is our interest? Our interest is blood, basically 7.4, and lemon about 2.3. See, these are all subjective. There is one activity 2.11 which is listed. You have to work out yourself activity 2.11, and one important thing out of that is saliva. Basically, in activity 2.11, he has asked what is the pH of the saliva before meals and after meals. So thing is, before meals it will be about 7.4, somewhere near the neutral range. After eating the food, what happens inside the mouth? The bacteria present would be able to act upon the sugar of the food particles, and they convert them into acids. So the acidity increases in the mouth, probably 5.5 and all, and therefore before meals the saliva will be somewhere around neutral range. After meals it will be acidic in nature, is one which is to be highlighted, and therefore I just took that example. You can work out activity 2.11, I think it is 2.11, and try to find out through internet or through practically in your lab and all the different pH values of the substances listed in that. Okay. Now I will also discuss some importance of pH in everyday's life here, and here what is the importance of pH in everyday's life is a question now. See, main highlights, these are the main points written, taken from your textbook. What is the importance of pH in our everyday life? In biochemical system, our body works within the pH range of 7 to 7.8. Meaning, for a proper functioning of a body, pH range is 7 to 7.8. 7 is neutral, 7.8 is slightly alkaline. Do you find acidic nature over here? No. So our body and most of the animals should be able to function within a narrow pH range of 7 to 7.8, and therefore it is very important that if there is a change in the pH, then it is a threat for our life, isn't it? The same thing is given in the next example. See, what is acid rain? The oxides of sulfur, oxides of nitrogen, whenever those mix with rainwater, they create acids, and the rain so formed is called acid rain. So due to acid rain, the pH of the water found in the lakes, rivers, etc. will decrease. If the pH decreases, acidic nature increases. In that case, it is definitely going to be a threat for aquatic life, is it not? Because most of the organisms, animals as well as our human body, works in a very narrow range of pH, which is 7 to 7.8. So if it is an acid rain, then definitely it is going to be a threat for aquatic, isn't it? That is what it is given. Now, what about the pH of the soil? See, most of the plants requires a certain pH range, maybe 7 to 7.6 or something like that. That means if the soil condition is acidic due to acid rains and all, then the crops will not grow. So how to treat that? Farmers will generally use calcium oxide, calcium carbonate or calcium hydroxide solution in order to treat the acidity. Why it is so? If the soil condition is acidic due to acid rains or etc., due to acid rains or some or the other reasons, then the soil will be treated with calcium oxide or calcium carbonate or calcium hydroxide solution in order to neutralize it or to increase the pH from acidic to the neutral side, so that the crops will grow once again. So which is also very important. What I mean by that is pH of the soil needs to be maintained, because crops require certain pH range, and acidic soil is also not good for certain crops, and basic soil is also not good for certain crops. It needs to be treated and maintained at a particular range. And there is an activity listed which you can look at the activity also. pH in our digestive system. See, our stomach releases hydrochloric acid. The gastric juice contains hydrochloric acid, the pH of which will be something around 1.1 to 2.1, which is highly acidic in nature, isn't it? And due to indigestion and all, sometimes due to indigestion, if the acidity increases, we find something like a pain in terms of the irritation. Basically, the increase in acidity of the stomach leads to irritation, and in that case we take certain antacids and all. What do? They neutralize it so that the pH is brought back to the normal conditions. And therefore pH of the digestive system is also important. One is due to indigestion. If the pH increases, then we feel acidic in nature, we feel irritation, and we can take antacids and neutralize it and bring it to the normal condition. What about the other function of HCl? Why it is so much of acidic? Why the gastric juice contains hydrochloric acid? The other function is it also kills the harmful pathogens, is it not? Microorganisms. So only under that acidic condition it would be able to kill the microorganisms, which could be harmful in terms of disease, causation of the diseases and all. And therefore in digestive system also pH plays a very important role. Yeah, these are some of the important points which I have discussed, and I request you to note it down in detail. Okay, so please note it down. Yeah, dear students, today we saw the most important part of the chapter, but more important than this is the chemical properties and the preparation and properties of the salts, and you can expect lot many questions from that. But these are all basics which you must know very well. because you may get 1 or 2 mark questions from this portion, whichever I have discussed. And make sure that you have noted all the points, and you go through the textbook also. Textbook plays a very crucial role over here, which is important. And one more thing I did not discuss over here, the dental caries. That is the pH of the mouth is also very important. That part I'll discuss in my next lecture. And today I would like to end the session over here, and in the next lecture I'll take up chemical properties of acids and bases, and one more lecture will be based on the properties and preparation and uses of salts. Okay, so see you in the next lecture. Thank you.